Okay. Good afternoon. We're here enjoying good grapes. I just understand from John, so that's good to know. Welcome, welcome. Please come on and sit down. And everybody out there who's online, we appreciate you showing up today. I'm Gene Giacomelli. I'm the director of the Controlled Environment Agriculture Center, where we'll be webcasting a fantastic presentation today. And I'd like to introduce Rafi Gruner to make the introduction of our special speaker. Rafi.
Thank you, Gene. So I'm going to tell a little story before we start because it was kind of a revelation to me. So last week, just before Thanksgiving, my wife and I flew to Denver to visit our son and daughter-in-law because she was about to give birth to their first daughter. And of course, everything went just fine. And home came the mother, the baby, and the placenta. And on the way down to the seminar today, I thought to myself, what is the endosperm, which is the topic of the seminar today? And I read online that the endosperm is a kind of an organ that surrounds the plant seed embryo to provide nutrition and support for the developing embryo. And I said, wow, that sounds just like the placenta. And indeed, Ramin agreed with me that there are homologous situations between the plant endosperm and the animal placenta. So I thought that was a fantastic revelation for me because I'm not a plant scientist. I'm learning as I go along, but my background is in animal physiology. So for all of you who don't know exactly what an endosperm is, now you know. So now it's my pleasure to introduce Ramin Yadegari, who is professor of plant sciences at the University of Arizona. And he was educated in California, got his bachelor's, master's, and PhD, and did postdoc, University of Irvine, University of California in Los Angeles, University of Irvine, and Berkeley. And then he came to Tucson after doing some other things. And he established a unique, in my estimation, a unique program here at the university in the plant sciences department. And that is the investigation of the title that's on the board, Understanding Seed Development Molecular Control Mechanisms that involve the crucial steps as the embryo, the seed embryo, develops and makes into a plant. We all know what seeds are, but I think most of us don't think about the seed being such a complex organism. And of course it is, just like in animal physiology. The other thing I wanted to mention about Dr. Yadegari is that he is the PI, the principal investigator of a consortium grant from the NSF to study this particular area. And the University of Arizona clearly has an enormous amount of strength in plant biology, plant sciences, and his consortium grant from the NSF is just one major indication of that. So without further, I'd like to introduce Ramin Yadegari, and he'll talk to us about the endosperm.
Can everyone hear me? No? I don't need that. Okay. Okay. Thank you very much. Thank you, Gene and Rafi and everyone, Austin, for invitation and organizing the meeting, the seminar. I've been here before. Since Rafi told a story, I can tell one story that I have. I actually was here back in late 1980s, it was 90, 91, as a graduate student at UCLA. We were studying seed development in Arabidopsis, actually embryogenesis in Arabidopsis. And at the time, one of our current colleagues, Ken Feldman, who had an initial landing here as a faculty member, had started generating a mutant population of Arabidopsis plants that was grown in the greenhouses right there. And so I actually sat in that greenhouse, head house, with a dozen or so of my colleagues for about a week. We stayed at a hotel down in Speedway. We would drive up, spend like 12 hours a day on the microscope dissecting embryos out of the seed pods of Arabidopsis, looking for mutants we were looking for. So that was in part, how I partially made my entree into studying seed development. And so I have very good, happy memories from the time I was here way back in Tucson. So today I'm going to talk about understanding seed development, but as Rafi said, I'm going to be talking mostly about endosperm development. I forgot, is it? No, okay, that's okay. About endosperm development. And so endosperm development is important because, as I'll show you, endosperm is the absorptive tissue of the developing seeds in flowering plants. It's important for embryogenesis as well as seedling germination. Before I get into my talk, oh, great, thank you. Before I get into my talk, I just want to give you an idea of what I'm going to talk about. And so some of the things towards the end, I might run out of time because there are some things that I want to cover in the beginning, knowing that this is a pretty diverse audience. I would like to make sure that you guys are sort of in line with the thinking that we have in the laboratory and the kinds of background that we are starting with. So first I'm going to tell you a little bit about what seeds are, what their origins, what their function and structure, and particularly endosperm. Then I'm going to talk about gene regulatory programs. What is gene regulation? Why is it important? And how is it important with respect to endosperm development? I will then talk about the approach that we are using, and again, sort of feeding off the basic approaches and techniques that are used in molecular biology. How do we identify spatiotemporal programs of gene expression that would help us to identify which are really the key goals of our projects, that is identifying or deciphering gene regulatory networks, or GRNs, that program endosperm development. And GRNs are not specific to the problem of endosperm development. These are basically gene regulatory networks it's a theme, a set of mechanisms that underlie development in all organisms. And then I'm going to show you some data. I'm going to highlight two recent projects that we have had. One earlier project in the large project, earlier set of experiments, and a later one as ways of highlighting what we have done, and then tell you a little bit about our future directions. Okay, so before I start, it's important to understand where we stand in terms of the processes that we're studying. And so where do we stand in the life cycle of the organism that we're studying? So it's important to sort of do a compare and contrast between the life cycle of plants and typical life cycle of animals. And first, and you can see almost immediately that we have a, that's okay. We have a much more complex component added. We have a more complex life cycle for flowering plants or plants in general as compared to animal life cycle. So you can imagine in animals, just like us, we have a mature structure, which is the body, our body. It's a diploid structure. And this diploid structure, in some structures, some organs within our body, mainly the sexual organs, we have development of formation of gametes. This is through the process of meiosis. These gametes come together through the process of fertilization to generate the new generation of the animal organism. And so you have these two key processes of meiosis and fertilization. And you're going to see these again and again repeatedly. Okay, no problem. It wasn't doing it earlier. I don't know what happened with the sun. It's okay. We can live with it. I can draw the versions for you on the board if necessary. Yeah. All right. So here in the plant life cycle, you basically have the basic series of events that are taking place: formation of a zygote, which is diploid; formation of the sporophyte, which is the vegetative structure that we're all familiar with, the trees outside, the corn plants in the greenhouse, for example, or tomato plants. And then within the specialized flower structures, you have meiosis occurring. And instead of producing gametes that occurs in animal system, you have formation of spores. And this is a direct product of meiosis. However, again, here's the complexity of the animal system, that is, spores undergo development to form a gametophyte. And this is a multicellular structure within which the actual gametes that participate in formation of the next generation through fertilization are present. Okay? So do you want me to try something else? Perhaps switch to a... I don't have a different adapter, but if we could... I could try it, or I can upload it to Dropbox.
I'll grab your flashlight real quick. Are you able to plug it in there?
Yes, I have it. Yeah. Okay. For those of you who are listening online, we're going to stop for a few minutes to switch our computer, which is misbehaving right now. Oh, we're good. Test. Okay, so if this one doesn't work, that's okay. I can just use this one. All right, so you have an added level of complexity in the plant life cycle, in which you have formation of the gametophytic generation. This is typically called alternation of generation, because in all plants you go through a generation which is haploid, that is, has half the genome amount of the genomic material or genetic material as compared to the typical diploid portion of the life cycle. Okay. Now, depending on which lineage of plants you look at, in some of the quote-unquote lower plants, such as ferns, for example, you have quite a large gametophyte, although it's not as large as the sporophyte. In other lower plants, you would have actually a switch where the gametophyte is larger than the sporophyte. And so again, you still have the same basic life cycle. You get the gametophyte undergoing fertilization. Products from two gametophytes come together to form a zygote. The zygote undergoes development, differentiation, proliferation to form a larger sporophyte. Again, that's a dominant plant you would see flowering plants. And then within specialized cells in that sporophyte, you have formation of spores through the process of meiosis. All right. So flowering plants have taken this to the next level. They're more complex in a sense that they actually are brought together, these processes, under the control of the sporophyte. They have actually reduced the gametophytic generation. So what you see in a typical flower in flowering plants or angiosperms, you have the formation of the female gametophyte, again, the female gamete generating portion of the plant enclosed within ovules that are enclosed within the carpels. Male gametophytes are, in fact, enclosed within stamens, pollen. Those are pollen grains within pollen sacs. And then their reproductive structures, they're all enclosed within a floral structure, which is relatively elaborate and has petals and sepals. And this elaborate structure very likely has evolved in order to allow pollination, efficient pollination to take place, and perhaps an underlying reason for the success of angiosperms. So if you look at the evolution of angiosperms or flowering plants, what you see is a set of major changes: reduction in the gametophyte size and complexity, increasing dependence of the gametophyte on the sporophyte. That is, the haploid gametophyte generation has come under near total control of the diploid sporophyte, which is now the dominant structure in the life cycle. You have the advent of heterospory that results in production of two distinct types of unisexual gametophytes. And more importantly, for the talk that I'm giving today, seed development was invented in flowering plants, and you have a double fertilization process in which a more, again, direct control of reproductive processes have brought under the sporophytic generation. Now, you can see this, again, in a sort of a conceptual way. I'm representing here maize as the example of a flowering plant or an angiosperm. This structure is the dominant sporophytic diploid structure. Within the flowers, the male and the female structures, you have meiosis taking place at some early time in the development of the flower. Meiosis produces two distinct two distinct spores, a microspore for male and a megaspore for female. These two spores undergo an elaborate set of developmental processes that I'm not going to talk about today to produce the male gametophyte and the female gametophyte respectively. Within the male and the female gametophytic structures, you have formation of the gametes, which are the sperm and the egg respectively, which then come together in the process of fertilization to produce the next generation, which starts with a zygote. It's a diploid structure, undergo elaborate developmental processes to produce the sporophyte, which then recapitulates the process again. All right. So I'm showing you various versions at different levels of quote-unquote zooming or magnification of the processes, so I can highlight the key aspects. Okay. So one of the key aspects that I would like to elaborate on is, again, the structure of the female and male gametophyte, particularly the female gametophyte. And the reason I'm doing this is I want you to see the context of seed development and endosperm development in the earlier reproductive processes that are taking place. This is the flower, a figure of the flower of Arabidopsis. Arabidopsis thaliana has been a model system for plant development for many years. And this this picture is actually quite close to the reality of what you would see. It has a simple flower structure, not a very colorful, elaborate petal or sepaloid structures. Sepals, petals, you have the stamens within which contain the pollen grains, which are the male gametophytes. You have the pistil or the carpel that contains the ovules. And if you look at the ovules, ovules are multi-structured, multicellular structures that contain the multicellular female gametophyte of the Arabidopsis, and it's typical of nearly all flowering plants. There are multiple cells here. I'm not going to bother with most of them. Just going to want you to know what they are. The key ones are the egg cell and the central cell. The egg cell and the central cell are the two female gametes that participate in double fertilization event, which is a hallmark of flowering plants. And so in this sort of brief animation, again, you're looking at the female gametophyte within the ovule from the side. A pollen tube reaches the ovule in the pistil during pollination. The pollen tube carries two sperm cells, two distinct sperm cells. One sperm cell, which is haploid, fuses with the haploid egg cell to produce a diploid embryo. Two copies. Just like the rest of us, we all arose as a result of embryogenesis from a zygote and more elaborate later developmental processes. The same in plants. However, unlike us, where we are results of single fertilization events, flowering plants have a double fertilization event because there is a second sperm that is delivered by the pollen tube. That second sperm finds a larger nucleus, the central cell nucleus. It's larger because it has the genetic material of the egg cell. It's a homodiploid nucleus. So the fusion of the sperm cell with the homodiploid central cell nucleus produces a triploid endosperm. Now, I'm showing this again because I want you to appreciate the complexity of not only early seed development and what is going to ensue in the following developmental processes, during which the seed will develop, the embryo within the seed develop, the endosperm will develop in order to support embryogenesis and subsequent developmental processes. What you have in a single seed, nearly at any stage of development, there are three distinct generations or genetic individuals, genetically distinct individuals. You have an embryo which is diploid. 2N means diploid. This embryo inherited one copy from the mother, one copy from the father. So it has a 1-to-1 ratio of the maternal and paternal genomes. Endosperm, however, arose as a result of that second double fertilization event. And because the central cell was homodiploid, you now have a triploid endosperm. And the genome ratio is 2-to-1 maternal to paternal. These two are coexisting in the developing seed, surrounded by a maternal set of tissues, seed coat integuments, which are maternal. They were diploid because it was the maternal, the sporophyte, that was responsible for formation of the ovule and subsequently the seed coat, and it's a diploid structure. So these three distinct, genetically distinct structures are interacting with each other during seed development, and I can tell you that current evidence suggests that embryo has to interact with endosperm, absolutely, otherwise the development of both would be affected in an abnormal way. And in fact, seed coat interacts with both endosperm and embryo. And it makes sense, because as the seed is developing, embryo is enlarging, endosperm is enlarging to some extent, and therefore the seed coat has to accommodate for the development of those developing filial structures, the results of the double fertilization event, that is the embryo and endosperm. All three are interacting with each other, but still we don't understand exactly the mechanisms of these interactions that are taking place. So I'm now going to come back to the larger, the overall process of the plant life cycle in angiosperms, and I ended up here with the formation of the seed. And so you look down here, what you have is a set of processes that are required for formation of the mature seed before the mature seed can undergo germination to produce a seedling, and the seedling would subsequently has to undergo development to form the mature plant. However, once the embryo formation has occurred as a result of double fertilization, you already have generated the new generation. So from this point on, you're looking at the subsequent generation. The embryo that exists in this mature seed is the same individual that will germinate and produce the next mature plant. What is going on in the seed? This is a sort of a snapshot of what is occurring in terms of the development of the embryo. That first product of double fertilization. You have a single cell zygote which undergoes a very, very definitive, stereotypic series of divisions to produce a pattern that is recognizable to almost anyone who's ever looked at any seed. No matter what it is, you would recognize the formation of the embryo through development to produce a mature seed where the embryo is contained within that seed. Now, once you have the mature seed, you're now at a point where you can pause development and reinitiate the process depending on the conditions. So whether it's moisture, temperature, light, all of those impinge on, depending on the species of plants you're looking at, and control the rate at which germination occurs and the rate at which the germinant would undergo development to give rise to a seedling. So this is the process of germination and seedling development, which really brings us to the formation of the mature plant. The embryos themselves, overall, if you look at the flowering plants, are of two types. The two major classes or lineages of flowering plants are monocots and dicots. And monocots and dicots, or eudicots, I apologize, eudicots, are identified as such based on, in part, the morphology of the embryos that they produce. Eudicots have two cotyledons, that is, the embryos contain two cotyledons, and when the seedling germinates, it has two cotyledons. And monocots have a single cotyledon. The overall structure is the same. The polarity of the embryos in both of these lineages is the same. You have basically an apical basal polarity. At the apex, you have the shoot apex or shoot meristem. At the base, you have the root meristem. The cotyledons are basically embryonic leaves that are important for supporting the germination of the seedling. And they have different functions depending on which lineage you look at. So for example, this is the process of germination in monocots. And so you have a radical that has to basically pop through the seed coat and find the soil or moisture in order to anchor the seedling and allow for germination to take place. You have a similar thing depending on the lineage in dicots, except as you know, again, you have two cotyledons in this case. So that's the difference between monocots and dicots. Eudicots sort of superficially in terms of the structure of the embryo. But with respect to endosperm, they show very different behaviors, or they show very different structures at seed maturity. Whereas in the case of eudicots, the cotyledons perform the primary role of storage compartment in the developing seed, as it's manifested by having cotyledons that nearly fully are contained within the seed or occupy the seed. The monocots accumulate endosperm to a high level, and in many monocots, particularly grasses, the endosperm is almost always the largest portion of the seed. So you have these two different strategies, two different kinds of behavior that you see in the formation of the storage compartment. That is, the primary storage compartment in the seeds in eudicots are the cotyledons, which are of embryonic origin, whereas in monocots and grasses, it's the endosperm, which is a filial structure, the second product of double fertilization. All right, so that's what you get at the end of the process. Why do we study endosperm? I can give you probably 6 or 7 different reasons, but there is a very good reason to study endosperm because it's required for proper seed development. It's required for proper seed development because at least in early stages of seed development, endosperm is involved in support of embryogenesis. This is clearly the case in eudicots. In eudicots, if you don't have proper endosperm development, you will not get a normal embryo. The same seems to be occurring in monocots. The endosperm storage compartment, particularly in monocots, is essential for proper germination and seedling establishment. Without it, you will not have proper formation of the next generation. From the perspective of our human society, perhaps more than 50% of our caloric intake, whether direct, us eating the material, or indirect through feeding of our animals, comes from the endosperm, from primarily the 3 major crops: rice, maize, and wheat. Therefore, there is a great importance in understanding how endosperm develops so we can take that information and apply it to modification and engineering of seeds for higher yield, higher quality of the food that we eat ourselves and feed to our animals. And also for support of a lot of industrial processes, endosperm is a big source of renewable resources in industry as well. Ethanol is a perfect example, although, depending on when you talk about it, it has different importance. All right. So this is the biological question that we're interested. What is the basis for endosperm development? How is it regulated? How can we take the information, apply it to modify it for producing better seeds, higher yield seeds? Before I get into the molecular analysis of endosperm development, I want to give you a little bit of a background in terms of what is it that we are actually doing and how we are doing it. Now, in order to understand basic biological processes at a molecular level, we need to understand how the genetic information is stored, but also interpreted to produce the ultimate products that perform the roles in the cells. So as we know, in nearly all organisms, you have this particular flow of information. DNA is read through the process of transcription in the nucleus, or depending if you're looking at prokaryotes, in the genome, into RNA molecules. RNA molecules are then translated. The information in the RNA molecules is translated into protein products that actually do the biological processes, carry out the biological processes. All of these processes can be regulated. So when I say regulation, I mean that depending on the cell type, depending on the time you look at it, depending on the context, you can control the rate at which the RNA molecules are transcribed in the process of transcription, how the RNA molecules are actually exported from the nucleus into the cytoplasm, and how these RNA molecules are translated, at what rate they're translated, whether they actually remain stable in order to be read by the process of translation, and even when you have the protein, whether the proteins are modified, are they degraded, if modifications allow for higher rate of activity for the proteins if they're enzymes, or they perform other roles such as structural roles. Now, from what we know in both prokaryotes and eukaryotes, the key process, the primary process at which most biological processes are regulated is the process of transcription. That is, production of that initial RNA molecule that is ultimately translated, or whether it's translated or not, is the key process in which regulation occurs. It's a process, it's a regulatory process that has the highest impact on the downstream biological biochemical processes. So I'm going to define the processes that occur in the nucleus that read the genetic material in the genome, open up the genome, the chromatin structure, to allow reading of the genes and transcribing them collectively as transcriptional control. Okay? If you sort of zoom in at that level, you could divide it up perhaps to 2 or 3 other processes. For the sake of this discussion, I'm going to only call them transcriptional regulatory processes. Transcriptional regulatory processes are mediated through the interaction of specific DNA binding proteins that recognize specific sequences near the genes that have to be regulated. Okay? So you have some genes that are expressed consistently, constitutively, in all cell types, in all instances, in all contexts. These genes are typically encoding products that are absolutely essential in all contexts. They're involved, for example, in breakdown of glucose. Without it, the cell will die. So those are called housekeeping genes. However, the majority of genes themselves are regulated very finely through these transcriptional regulatory processes by and large. And again, the regulation occurs through binding of specific DNA binding proteins that find specific sequences upstream of genes and dictate whether those genes should be transcribed or activated or not. So there is a give and take. The DNA binding protein would recognize a particular binding site, and these recognition processes are very specific. That is, there are proteins that have evolved to recognize very specific short sequences of DNA upstream or near the genes that are regulated. The give and take is that once that binding occurs, that binding would then allow for binding of a much larger complex that is actually performs the enzymatic function of transcribing the gene into an RNA molecule, which then will go on outside the nucleus and be translated into a protein. So this is a very simple scenario that I'm presenting here. It's probably the simplest scenario, and like everything in biology, probably doesn't really exist because it's too simple. But perhaps in some prokaryotes, in bacteria, you might see this instance. What you see most of the time in eukaryotes is a highly complicated process. First, binding, again, recognition. These sequences can be called enhancer sequences. If they're particularly involved in recruiting proteins that are involved in activation of genes, sometimes they're called repressive sequences because they're involved in recruitment of proteins, transcription factor proteins, that repress the nearby gene. Regardless, there has to be a recognition, binding of the protein, and that binding would then allow for assembly of a complex of proteins that contain perhaps, in some cases, dozens. There have been reports of some complexes, transcriptional complexes, as many as 65 proteins have to assemble in order for a single gene to be transcribed. And that's only when you have a single enhancer sequence involved here. You can have multiple enhancer sequences involved in many of these cases. So the focus is going to be here. The binding of the activator proteins or repressor proteins, or what I'm going to call transcription factor protein in a much more colloquial fashion, is the key determiner of whether you're going to have the downstream genes or nearby gene actually be activated or repressed, that is going to be expressed or not expressed. You can actually decipher the nature of these interactions by simply doing a lot of biochemistry and molecular biology I'm going to describe to you briefly. This is an example of a very advanced case using sea urchin embryos. And these are cells called skeletogenic micromere cells that occur in a very particular location in the early developing stages of sea urchin embryo. These gene activation processes do not occur as single genes. Most of the time, you have to have multiple genes, many times multiple transcription factor genes activated in a particular sequence in order to allow for the downstream genes that actually encode important biochemical products to perform the function. So in this case, these micromere cells that are labeled as red, they are activated ultimately, or they're expressing a particular set of genes, and they ultimately will give rise to micromeres during development. That is the sequence of these cells undergoing changes that you see here to produce the skeletogenic lineage in the sea urchin embryo relies on the sequence of events that take place both in space and time with this particular transcription factor protein being activated. And in many cases, we're going to show these. You can show these as simple on and off logical steps. On produces a protein that surprisingly acts as a repressor for a downstream gene. In absence of activation of this downstream genes, these other 4 genes are on constitutively in those cells. And so the expression of these 4 genes determines how these red cells go on during development to produce these skeletogenic cells. In contrast, everywhere else in the cell, in the embryo, you have the activation of this gene, which in turn, again, represses the downstream genes, which prevents the rest of the cells of becoming skeletogenic. Okay, so you have a dual situation. Activation of this gene is required for expression of these downstream genes simply because it represses an intermediary gene, whereas in this case, activation of this gene acts as a direct repressor of these downstream genes. So you can see what level of complexity we're talking about. This is another example of a circuitry which is actually simple. Simple. It is a circuitry that determines the top and bottom of the fruit fly embryo. When the embryo is laid down, it has to be determined through intrinsic regulatory processes, exactly this network, what side is going to be the back of the fly and what side is going to be the stomach of the fruit fly. And so this circuit took probably 20 years to figure out. All right? So we're talking about a complex scenario, and you can represent it in these simple diagrams. Okay. So coming back to the biological question of interest, we are interested in determining the gene regulatory networks that control early endosperm development in maize. And so this is double fertilization in maize. It starts again with the pollen tube delivering two sperm cells. One fuses with the egg cell to produce the embryo. The other one fuses with the central cell nucleus to produce the endosperm. Surprisingly, in both dicots and eudicots and monocots, early endosperm development is very similar. Okay, they might be different in terms of the size of the ovules, the size of the gametes, but the patterns are actually very similar. All right, so that is the process. However, this process hadn't been really studied very well in the accessions of maize that we're currently using to study all aspects of maize development. And so our collaborative group, in the first set of funding that we obtained back in 2009, started to look at first establishing the patterns of development before we could even start looking at genes that are expressed. And so this is a collaboration with a laboratory at Central Michigan University, Joanne Dannenhofer, who actually happened to be a postdoc, previous postdoc at University of Arizona. And so she and her students basically very painstakingly prepped early developing ovules and seeds from maize, and over time using both fixing, embedding, sectioning of material, as well as using whole mount confocal laser microscopy, determine the patterns of development in early endosperm development, in early maize development, maize seeds. And so this is basically what we're looking at. We're looking at the first 12 or 15 days after pollination. What in the top row you're looking at hand sections from the ovules before pollination, 1, 2, 3, 4, 5, 6, 8, 10, 12 days after pollination. But just to orient you, at these early stages of development, you can barely see the embryo, you can barely see the endosperm, but here on you can see the endosperm developing. Here are the embryos, and at 12 days, the endosperm nearly has filled the entirety of the developing ovule or developing seed, and the embryo has gotten big as well. Now there is an interesting pattern that is occurring in early endosperm development in maize as well as most flowering plants. And the pattern is one that is called synocytic. That is, rather than having cells forming that undergo mitosis to produce daughter cells and undergo mitosis to produce daughter cells, early endosperm development in nearly all flowering plants has an early period in which cells do not undergo division, nuclei undergo mitosis, and you actually have a cavity, a cenocyte, a multinuclear cavity where endosperm nuclei are positioned outside the periphery of this developing seed, and later on, through some mechanism that we don't understand, they undergo cellularization in nearly coordinated fashion. They ultimately all undergo cellularization and fill up the cavity of the seed with a cellularized endosperm. So that's why you see this cenocytic period. The cenocytic period occurs in the first 3 days after pollination. You have a programmed cellularization occurs that is finished by about 5 days after pollination. And then after that, the endosperm is all cellularized, and all divisions, all proliferative processes are occurring very rapidly through standard cellular mitosis. Okay? So we spend a lot of time, when I say we, it's primarily actually Joanne's group, spend a lot of time figuring out what the stages are. It's very important when you start using a system as a developmental system to actually be able to reproducibly generate the same stage of development in the same location under perhaps slightly different conditions because you can't avoid it, but also be able to generate the same stages in different geographic locations. And so one of the challenges that we had here was that plants in Arizona in the greenhouse grow about a day and a half to 2 days faster than plants in Michigan or Utah, for that matter, where our other collaborator was located. And so we ultimately, the people in Michigan, Joanne's group, they use these numbers that are based on the stages of development, that is days after pollination in Arizona, but they gave them the numbers that are absolute numbers to denote the stage of development that matches what's going on in Arizona. So that was one of the challenges that we have to deal with. So here I have sort of tried to summarize what happens in the first 12 days after pollination in the developing seeds in maize. And so what I showed you earlier was the little bitty developing embryo and endosperms in much larger seed ovule structure or early developing seed. What this is, what the blue here is actually a structure called nucellus, whose function we still don't understand, but we believe that nucellus is actually an absorptive tissue that supports early development of the seed by supporting the developing endosperm. So endosperm being an absorptive tissue for support of embryogenesis and seedling development, nucellus, at least early in seed development, is likely an absorptive tissue for the support of endosperm development. In my lab, the joke is that nucellus is an endosperm for endosperm. So that's probably what's going on here in these early stages. But soon, within the first 6 or 7 days after pollination, the nucellus is completely absorbed by the developing kernel, by developing seed. And so what you have here, again, you have the pericarp structure, which are the maternal kernel structures supporting the developing embryo and endosperm, which are the filial products. Here again, the sinusitic growth period, followed by cellularization, and then you go into mitosis to increase the bulk of the endosperm, as you can see, after cellularization. And that's why you need very rapid increased levels of mitosis in order to generate this bulk of endosperm that ultimately undergoes cellular differentiation. That is, there are portions in the endosperm itself that undergo processes to specialize them to perform particular roles. And I'll show you what those roles are as best we can decipher. And to prepare endosperm for its primary role, which is to accumulate storage products, carbohydrates, starch, and proteins. And I'm gonna probably mention that a little bit later. So in order for us to understand endosperm development, we decided to take an approach that was enabled partly through the development recent technology. Now, as a developmental biologist, I have used the standard approaches that have been used over the past 100 or so years, pioneered by people working on bacteria and Drosophila, that is looking for mutations that change some aspect of development, some characteristic. Mutations are changes in genes. Changes in genes can be identified reproducibly. They are maintained. They're heredited. So they're consistently inherited through multiple generations. One can actually map and ultimately identify the genes that are mutated to produce a particular phenotype. That phenotype is informative of the function of the gene in the normal or wild type situation. That has not been very useful, frankly. It has been very useful, actually, for studying many basic processes, deciphering many basic processes in plant development, plant growth, plant physiology, plant response to hormones and such, using particularly Arabidopsis and to another degree maize. But in our case, in the case of seed development, it has not been very useful in deciphering the processes that we are interested at a very basic fundamental level in maize, or for that matter, for most flowering plants. So we decided to take a different approach. In the case of identifying mutants or using genetics, we're looking for is mutations in a single gene that produce a dramatic phenotype. Those changes are usually likely occurring in genes that perform very important roles. There are examples, as I mentioned, in other processes where mutation in transcription factor gene that regulates, for example, ethylene response can be readily identified based on the phenotype and the available tools of genetics and molecular biology Arabidopsis. That wasn't the case in maize, hasn't been the case for most of the genes that we're likely involved in seed development for a number of reasons. And so as an alternative, we decided to use what I call the bottoms-up approach. That is, try to identify basically all the genes that are expressed in any cell type that is responsible for the formation of endosperm or any structure in the seed. Again, the idea is to identify every gene that is either expressed or not expressed in development in order to understand the extent or decipher gene regulatory processes. All right, so the approach is actually pretty straightforward, or the concept is pretty straightforward. The concept is the following. The concept is we want to identify bins or groups of co-expressed gene products. These bins can then somehow be used to determine what genes are expressed at a given time together via activity of one or more transcription factor genes. So the idea is that each bin should contain the genes that are expressed at that time or in that location in development. And it should also include the transcription factor genes that are expressed at the same time that are presumably responsible for activation of those downstream genes. So you're going to hear this over and over, downstream genes, genes that are activated or repressed by one or more transcription factor proteins. Okay? So these bins, I'm going to refer to them as co-expressed modules or co-expressed genes. And you can imagine these co-expressed bins can be obtained from collection of proteins that are made from a given cell type, a given time in development. They can be made from perhaps the collection of all the lipids that are produced, all the small molecules that are produced at a given time. Currently, our technology allows to identify the co-expressed mRNAs, the RNAs, the intermediary molecular genetic material, much more reproducibly with a higher power, higher depth than any other gene product. So that's why you're going to hear a lot about RNA sequencing. That is, we collect RNAs from specific stages of development or specific times in development, and we're going to use the available technology, which has evolved enormously in the past 10 to 15 years, to sequence, and I'm not exaggerating, we can potentially sequence every mRNA that is produced at any time in development. Now, as an example, Arabidopsis has about 26,000 genes. Maize has about 5,000, who knows how many genes actually. Every gene is expressed at a different rate. Some would produce only one mRNA molecule, maybe 0 mRNA molecule at a given time. Some can produce as many as hundreds of thousands of mRNA molecules. Our technology today allows us to basically profile the entire spectrum all the way potentially to a handful of mRNAs that are produced at a given time from a given gene. Okay? So that's what I mean by depth when we're talking about it. That's why it's called deep sequencing now. All right. So I have perhaps impressed you with the idea of sequencing RNAs as a key inroad into identifying co-expressed genes that would somehow allow us then to establish interactions between transcription factor genes or proteins and their target genes. Okay, so our first approach, what I call the first pass experiment here, was basically to take stages 0 through 12, bracketed, and what I'm showing you here is hand-dissected kernels, which are stained with toluidine blue, just to show you what the outlines look like, and iodine potassium iodide, which highlights starch, to show you what they look like. Okay? And again, we could reproducibly, before any of the people in my lab even touched the material, before they even sent the material to be sequenced, before they did the first RNA extraction, they had to prove to me that they could produce these stages in a reproducible fashion at various times in the season from various plants. So that was very important. So we could repeat this if we needed to, and we had to repeat it. So anyway, so we collected whole kernels, and we collected hand-dissected endosperm from these stages. And we made RNA from these, extracted RNA, and we sent the RNA to be sequenced using deep sequencing approach. Now, the idea was that we were looking for genes, that is, presence, absence of RNAs, that are expressed at one stage of development and perhaps not at an earlier stage or not at a later stage. Okay? We were looking to identify how changes in gene expression over time can be extracted from this data. So we extracted RNA from those stages. We applied a particular kind of algorithm that's been available at the time. It's called StepMiner, doesn't matter. It will be looking for, a program would be taking our RNA sequence data. And RNA sequence data is not just sequence. It's not identification of the nature of the RNA or the genes. It's actually counting the number of RNAs that are produced from every gene. So it's giving us a quantitative output of the expression of genes during development. So the program takes the counts the number of RNAs for each gene at any stage and tells you whether there is a blip, up blip or down blip, an up down or a down up blip. Okay? In order to find programs of gene expression that would be associated with those changes. And so this is data from that analysis. These are, the red and blue are average graphs for those gray lines that you can barely see. Those gray lines is the first number. So for example, at this stage, we had 523 genes that were identified to be upregulated at 6 days after pollination. Upregulated at 6 days after pollination, meaning that 4 days after pollination, gene expression was low. All of a sudden, at 6 days, 523 genes are activated. Okay? And then genes that were downregulated correspondingly. These are two separate sets of genes. Okay? So these are the kinds of data that we could extract out of that analysis. We identified in total more than 7,600 genes that show this up or down, unique up and unique down patterns of expression. Among those, there were 100 transcription factor genes. So just think about it. So this suggests 100 transcription factor genes are upregulated or downregulated. And that up and down regulation correspondingly is responsible for up and down regulation of 7,600 genes that are either directly or indirectly regulated by those transcription factor proteins. Now, yes? So this column, these are upregulated genes. So they're up at 2, 3, 4, 6, 8, 10, 12. Same exact stages. Absolutely. And mind you, there are thousands of other genes that don't change patterns of expression that are also expressed. Yeah, sure. And so we then looked to see what these genes encoded. Because we had the sequence, we could actually take this sequence, conceptually translate it to what the mRNA would likely encode as a protein. And what we saw was very interesting. As more or less sort of expected, at these early stages, there are a set of genes that are encoding cellular processes that are associated with amino acid synthesis, metabolism, RNA synthesis, all of these processes that are expected to be occurring at early stages of endosperm development. A little bit later, within 2 days, these processes, first set of processes are expected to be responsible for formation of proteins. The second set of proteins, set of genes within 2-day period, are primarily responsible for carbohydrate synthesis and accumulation. So you have a sequential activation of genes that are responsible for first producing lots of proteins that are likely involved in establishing, all the structures of the cells that are differentiating or proliferating in the endosperm, responsible for accumulating lots of proteins, and then behind it, set of genes that are activated in order to allow for carbohydrate and starch synthesis to occur. Okay, so sort of makes sense. I'm going to go over this very quickly. RNA-seq, again, is the approach that was used to identify those RNA sequences and identify the gene expression patterns. So on this left side, I'm showing you examples of 3 genes that show this upregulation. These 3 genes are not any other kind of genes. They're actually transcription factor genes that we glean out of this data. And then what we had to do, again, people in my lab had to convince me, and then we had to convince the reviewers of the manuscript that this data is actually not bogus, it's not artifactual. So what we did is use a different independent method for quantifying the levels of mRNAs in the material. In fact, we collected separate sets of material as biological controls, and we showed that, in fact, independently we can reconstitute the same patterns as we see with RNA-seq. So that allowed us to feel very comfortable about this set of data. Here I've brought together many of the transcription factor proteins that we analyzed in our data set. And I wanted to sort of introduce you to the concept of looking at levels of expression using a heat map. Okay? So this is a graphic representation. Rather than showing you, these bar graphs, which is going to give you a headache, there is a new method, and we're moving into using heat maps because we now have to show you. I'm just showing you maybe a dozen genes, but I'm going to show you examples of cases where you want to visualize the expression of 10,000 genes on a single screen, on a single piece of paper. And so heat maps become very useful in that case, and that's what we're doing here. So these genes are not highly expressed at early stages of development, and they're upregulated, and that upregulation is visualized using this more purple redder color. Okay, so it happened that 2 of these transcription factor genes are genes that have been identified previously. So again, it helped us believe a little bit more in the data that we had generated. Okay, so I showed you this temporal program of gene expression. There is an associated change that is occurring with respect to cell differentiation and changes at the cellular or spatial level. And so as early as 6 to 8 days after pollination, as I mentioned earlier, there are actually cell types that can be recognized within the endosperm. So I know you guys have eaten maize kernels. It looks like sort of a homogeneous type of thing. It is not homogeneous by any stretch. Nearly at any stage of development you look at it, there is some specialization that is going on. So as early as 6 to 8 days after pollination, you already have an aileron layer. Aileron layer is the outer cell layer of the endosperm. It is the outer cell layer that ultimately is responsible for controlling the use of resources of endosperm during germination by the embryo. There is a starchy endosperm, which is the bulk of the endosperm responsible for starch accumulation. Not very, very imaginative names. That's just the way they are. Embryo surrounding region, again, a region that surrounds the embryo. These are all specialized. They can be recognized based on cellular morphology to be different cell types. There is the basal intermediate zone. This is actually something that we discovered, and we just applied that name to it. It's a different region. And then there is the basal endosperm transfer cell layer. This is actually a very important cell layer in endosperm development because this is the cell layer that is sitting right above the maternal seed coat cell types, and it's responsible for uptake of sugar and metabolites from the underlying maternal tissues into the endosperm, facilitates movement of sugars, and ultimately accumulation of sugars and transfer of sugars into starch in the process of accumulation. Now, there are subregions within the starchy endosperm, and so you can see already at this early stage, it's a highly specialized structure. So this is something that we've been interested in, and so we decided to take those genes that we had analyzed, those genes that we had identified via their temporal patterns of expression, and see where they're expressed during endosperm development. And so here's a new method that is used. This is a method that has been around a long time. but new in the sense that we're using it first in this talk, and that is you can actually visualize the areas, the cell types that express a particular gene using a method that can allow you to visualize where the RNA is located. It's called in situ hybridization method for mRNA. So you generate some sort of a probe that can recognize that particular mRNA in those particular cell types. And what you're looking at is basically the expression, the accumulation of RNAs for different genes in different parts of the endosperm. And so you have accumulation of these different genes in different cell types, and it's sort of shown in this cartoon. Again, for example, in the case of BIS was identified by the virtue that a number of genes were basically expressed in this portion of the endosperm that had not been recognized before. Okay, so we did this analysis, and so we now have two sets of data. We have a temporal set of data, we have a spatial set of data, and we decided, why don't we put it together? We put it together, and so what you can see are these cell types. These are genes. Don't worry about the numbers. No one can remember these things anyway. They're just for identification purposes. You have in columns, you have cell types or compartments of the endosperm. In rows, you have genes. This part of the chart shows you the temporal patterns that I showed you earlier. This part of the chart shows you the exact expression patterns. I'm sorry, this part shows the spatial patterns of expression, and this shows the temporal patterns of expression. And so what was interesting to us, was that genes that are expressed in aileron and then in basal transverse cell layer and BIS are expressed earlier as compared to genes that are expressed later, and these genes are primarily in the starchy endosperm. So there is a sequence of activation of genes that goes along, very likely, with the development and specialization of the endosperm as the endosperm is developing within the developing seed. So this is sort of a story that we developed early on in the process. I'm running out of time, so what I'm going to do is I'm going to basically give you just a hint of what came out after this, which was, again, I told you we're interested in cell differentiation. How do we get at this process? And so the way we got at this process, which turned out to work actually, otherwise I would not be talking about it, is that we determined that we're going to identify genes that are expressed in specific cell types in the endosperm, and this was not a trivial issue because this is a very small kernel, small seed. And so the way we approach this is by using a microscopy technique called laser capture microdissection. There's a particular kind of microscope. You can go in, you have to prep the tissue appropriately, you have to section the tissue, you put it on the microscope platform, you bring it up on the computer, and then you point to the portion of the tissue or the endosperm or the seed that you want the laser to cut. You draw it around, that in this case is basal transverse cell layer. You cut it, you basically draw a thing on the monitor, and the microscope goes in with its laser, it cuts it out, drops it into the tube for you. Okay, it's small, tiny amounts of material, but it contains the RNA still for those cells. So you're isolating cells from other cells, you're purifying cells, and so that allows basically purification of these different cell types via this method. And so we carried out a series of experiments. I'm going to bypass this information to tell you that we ultimately were able to produce the bins. Now, here bins are not temporal bins. They're not based on times after pollination. There are spatial bins, different cell types of the same stage of development. We just want to understand what are the genes, what are the gene regulatory networks that are responsible for making a basal portion of the endosperm be basal transverse cell layer. How would a starchy endosperm become a starchy endosperm portion of the endosperm? So these bins, or spatially co-expressed mRNAs, when we analyze for expression, we submit it to analysis using RNA-seq through a pipeline, put it through analysis through a process called WGCNA, doesn't matter what that is. It was able to bin it for us, And so what you see are each blue line is a single gene. Some of these have as many as 1,000 genes in each bin, some 500, 600. And what you see is commonalities, upregulation or predominant expression in a particular cell types. The cell types are shown in the bottom. Here is an example. This is the bin for basal transfer cell layer. The genes show all sorts of patterns of expression all over the place for other cell types. Only one cell type. They're all upregulated. They're all activated. Okay? So this module called Module 18 turned out to be very useful for us. Here I'm showing you a statistical treatment of this. Again, I'm going to bypass this. To tell you how we got at the network, ultimately, in Module 18. Again, the key thing for us was to be able to connect a co-expressed transcription factor protein to the genes, all of which showed the same pattern as exemplified by Module 18. Expressed everywhere else at very different levels, but they're all upregulated in module in BETL. And so what we did was, again, through using a series of bioinformatic analyses, we identified the putative sequences upstream of those genes. And in fact, in this bin, there were 600 genes, more than 600 genes. Many of them contained this sequence at a high frequency. And when you look at the literature and look at our transcription factor genes that are expressed in the same bin, identified and included a transcription factor protein called MRP1, have been described previously to be involved in differentiation of the ETL. Okay? The same bin where this RNA came from. And in fact, we identified additional sequences to which MRP1 would potentially bind. These sequences had been previously identified. We added a lot more sequences to this. And then the next step was to somehow validate binding of MRP1 to these cis motifs that are presumably involved in mediating interaction of MRP1 with the target genes and therefore activation of these target genes. And so we used, through collaboration with Gary Drew's lab, University of Utah, we used what's called a yeast-1 hybrid assay. That is, you can drive MRP1 using a yeast regulatory sequence. The presence of that transcription factor protein in yeast would then allow recognition of whatever motif you want to test. And so we tested all of those motifs that we identified bioinformatically and showed that, in fact, the yeast grows well. And so ultimately, we established a small gene regulatory network that has MRP1 as a node, a transcription factor protein that can interact with 4 distinct cis regulatory motifs. There is, again, sequences upstream of genes that it's activating, and it helps to activate 90 or so genes in that module. Now, mind you, the module 18 that I showed you, that bin contains more than 600 genes. So what's going on with the rest of them? Well, we don't know. We're working towards that goal. One of the issues that we have is that MRP1 is not unique transcription factor protein in that bin, in that module. It includes 5 or 6 other transcription factor proteins. And it happens that these are all belong to the same family. And they're all highly related, evolutionarily speaking. Okay? So they very likely arose as a result of a recent gene duplication in the maize lineage. And so they're all likely involved in regulation of BETL gene expression. So we are now trying to understand how the BETL gene regulation, or genes expressed in BETL regulated, are they regulated by distinct MRP1-like genes? Are they regulated in overlapping fashion? These are the kinds of questions we would like to address. Just to end, I'm sorry I'm going a little bit over time. The early spatial temporal program is what allowed us, identification of these spatial temporal programs allowed us to decipher the network. And so we want to go earlier and earlier. And so we are now working on identifying genes that are expressed immediately after fertilization within 1, 2, 3, 4 days after pollination in the early developing endosperm, when it's just a very small, tiny amount of endosperm. We would like to know what are these transcription factor proteins binding to throughout the genome. What are the processes that are responsible for that? Is there a conservation of the binding of transcription factor protein and activation of the targets among maize, sorghum, and other monocots and grasses? These information would be important in order to figure out a way to engineer size, because based on the current data, increasing the rate of early endosperm proliferation is directly correlated with an increased seed size in multiple plant species. Okay? So it is likely that if we can regulate this process, we could regulate seed size. Another frontier that we are now beginning to work on is understanding how labile these early developmental processes are to stresses, including drought and heat. And particularly drought is of great interest because of obvious reasons. It's likely that endosperm is a major sensor of drought stress in early seed development, because in many plants, early stress causes seed drop. And so we would like to understand what is it in the endosperm that is able to recognize that. Okay, finally, I would like to thank the people who actually did the work. Again, as Rafi said, this work was really funded by, now we are in the second iteration of a plant genome grant that has funded us for the past 6, 7, 8 years. The people who did the work, Shanshan Zhang, I didn't really show a lot of work from her, a lot of data from her. The primary work had been done by Guosheng Li, by Junpeng Zhan, and I have been lucky to have a good set of undergraduate students working in the lab. Our collaborators, Gary Drews, University of Utah, Joanne Dannenhofer, Central Michigan University, Phil D. Kraft at Iowa State, and Jiangpeng Wang was a colleague of us at University of Arizona in my department, who decided to move back to Beijing a few years ago. And then I've been the luck of having a good set of collaborators as well for the past few years. Sorry for going over time. Thank you for your attention.
Thank you, Ramin. Sorry for our technical problems that force you to go over time, but let's see if Jack has a question. Brief. You need the microphone. Yes.
Dr. Mendel, when he looked at corn, said, we're learning something about genetics. And I'm visualizing that each kernel is giving us information by color about endoderm. And so I'm maybe totally wrong in the way I'm looking at your model, but you're telling me about thousands of genetic steps that go from a very early stage to an ear of corn with different colored kernels. Tell us about what's really the aspect of that which we now can use as our forefront for gene to basically bring this model into a closed environment and maybe go away from soil and then understand a lot more about the mechanics of what's been happening and what can happen in the future.
Actually,
where'd that question come from?
The latter part of your question. So the first part, I mean, I think it is just like any flowering plant seed. Each seed is an individual, and so yes, they arise on the ears of a distinct genetic individual, but each of those individual kernels contain an embryo. The endosperm behave totally independently from each other, and so you can actually see different changes in differences in behavior, whether it's endogenous behavior or changes to the environment on individual kernels. To answer the latter part of your question, actually is something we just talked about before the seminar started. That is, one of the things we are interested in is understanding the effect of exogenous changes on kernel development. And so, in the field, even in the greenhouse, using a standard greenhouse condition with the soil and things like that, we cannot necessarily reproduce response to drought, response to heat very well. And so I think there is a great potential for using controlled environment agriculture type of setup, and perhaps even developing it further to establish a uniform, reproducible condition, which then could potentially address questions that you address, even for changes with, from plant to plant, and also potentially even from the same era.
Excellent. I like how you brought that right around. Thank you very much. Unless there's a real burning question, we're pretty late here. I think we'd like to call this to a close. I think everybody is shaking their head. We can always find Ramin, email. We know where he lives. We can get the questions to him. So I want to thank everyone for staying and participating, and Ramin for your time and efforts for coming out here on this beautiful Friday evening in midwinter in Tucson.
Thank you
very much. Thank you again.